So there's the cycle. Interphase, prophase, metaphase, anaphase, telophase, interphase, back around. Now those same stages occur in the other type of division, meiosis. Offspring are a genetic mix of the parents. How does that happen? Well, the parents each have two complete sets of codes. Now the codes, the two codes may vary a bit. They may be different alleles. They may actually produce significantly different proteins, but the gene is essentially for a protein that's supposed to do something. Sometimes it does something fairly different, but. So you've got one set of genes and a second set of more or less duplicate genes. You're only going to send one set onto your offspring, unless you're reproducing by yourself, which, remember, you can do sexually. But that means that you need to produce at least a nucleus. And again, this is actually nuclear production rather than cell production. At least a nucleus with only one set, a complete set, but one set of genes, one set of chromosomes. Very often packaged in a cell, because remember, the most common form of sexual reproduction is actually sperm and egg cells, is actually male and female. But that's not the only way to do it. And in some organisms, haploid nuclei, single set nuclei, are exchanged. So what we have here are the differences. Parent cell. I'll walk you through this slower, but I'm going to go fast on this first one. Evolution tends to be pretty conservative, especially in really important areas like reproduction. So you're getting ready to make some sort of production system. What do? We make copies of all of your DNA. We don't need to make copies. We're trying to divide sets. What are you making copies for? Because that's the way that it works. And that's what's going to happen. In both cases, DNA is duplicated during interphase. So the problem with meiosis is that now you had two sets of chromosomes, now you got four sets of chromosomes. So you're going to need to make four cells or four nuclei. And so one of the first things about meiosis is that you're not producing two, you're producing four. And the easiest way to do that is to do it in two steps as opposed to a single step. Here's the second step, right? Here you've already produced what you need to produce. Here, you're halfway there. So meiosis is a two-step division process. Could be nuclear division, could be cell division, could be different size cells if you're making egg cells. But you're basically going to make four as opposed to two. What happens is the first division in meiosis, here's meiosis, is called Meiosis I. Although it's typically written as Meiosis I, but I don't know that I've ever heard anybody call it Meiosis I. They call it Meiosis I. That's not a big deal. Here, Meiosis II is Meiosis II. Again, I don't think I've ever heard anybody call it Meiosis II, but that's the way it typically is written. Because we're biologists, so we're only happy when we're confusing the world. So here's where the big difference is. During metaphase of mitosis, you just get your chromosomes lined up kind of haphazardly, because you're going to pull the copies away from the copies. The first division in meiosis is to get the sets away from the sets. Now, how do you do that? Well, you take the matchers of each set, like in humans, we have 1 through 22 and sex chromosomes. During sperm production and eggs production, at some point, during prophase, and I will tell you, I have no idea how this works. Again, something I've tried to look up, and somebody's got to know the mechanism, but I haven't been able to find it. Number two chromosome, they wind up next to each other. Number 22 chromosome, they wind up next to one another. So the matched pairs are paired up during prophase, and they're lined up together during metaphase. This takes a while. If you're the first pair in there, you're right up next to each other. And what happens is, imagine you're next to somebody in an elevator and you brush them with your arm, and when you release, part of your arm is now on them and part of their arm is now on you. If you bumped at elbows, you now have their forearm and they have your forearm. If you bumped your elbow against their shoulder, boom, then their shoulder has your forearm and your forearm has their upper arm and their forearm, but you also have your upper arm, right? Because you only bumped your elbow, you still have your upper arm. This movement of one chromosome to another is crossing over. And when we get to genetics, this is what screwed up people who were just figuring out how linkage worked, when they found that it isn't always reliable, because linkage is genes that are riding on the same chromosome, alleles that are riding on the same chromosome, that should be passed on as a package because they're physically connected to one another, except when they're not. Sometimes they swap over from one to the other. Crossing over, it introduces another level of variation. But the thing is, sometimes the swapping isn't equal. Like the whole idea of, so I bumped your elbow at your shoulder. So your shoulder from there down is now on my lower arm, but I already had an upper arm, and now I have your upper arm. This is a process of gene duplication. Unequal crossing over can put extra copies of certain genes on the recipient chromosome. Because one of the big mysteries of genetics is, okay, I'll take that mutations can produce a new function in a protein. What happens to the old function? Wasn't the old function important? How do you get a new function and not lose the old function if all you're doing is changing the gene code to produce a new protein? Well, what if we had extra copies of those genes? That turns out to be unequal crossing over. And so you've got the original function of the original gene, and now you have a second copy that mutation can actually alter, and you don't lose the original function. If you look at rattlesnakes, their gene for digestive enzymes is very closely linked to their gene for venom. And they're very homologous. They've got very similar sequences, but one of them produces a neuroparalytic protein, the venom, and one of them produces a digestive enzyme, which breaks down the tissues of the prey animal, starts to at least. Spiders, you have another thing. And apparently, probably the digestive enzyme was the original gene, and the venom was an add-on change in a duplicated gene with unequal crossing over, because you kind of need the digestion before you absolutely positively need the venom. Venom's useful, but not as useful as being able to digest stuff. But this also explains why when you go down family trees, you tend to find more and more DNA and more genes. It's just like, where the hell did the new genes come from? Where's all this extra DNA coming from? So Meiosis I, you can see if you can make out the colors. And what separates in anaphase in Meiosis I are the sets. So number threes are up next to one another. One number three goes this way, one number three goes that way. And so now you have just one set in each of these cells. The problem is you started out by making duplicates. So this is a set of duplicates connected to duplicates. You got to get those separate. That's Meiosis II. Meiosis II looks like mitosis. Chromosomes get in the middle, the strands separate, they get pulled apart. Great. The real difference is the number of chromosomes you're starting with. in humans, you start with 46. Now each of these has 23 duplicate chromosomes. These wind up with 23 single-strand chromosomes. So that is the major difference between mitosis and meiosis. Meiosis II looks like mitosis, it's just the numbers are different. Now, I don't know that we've covered this semester, so we're going to get into this. There is a difference between production of sperm cells and production of egg cells. Remember, egg cells specialize in giving your offspring food to get started with, so that they can develop and get to a point where they can pick up nutrients themselves. So egg cells are large, they're expensive, you don't make as many of them compared to how many sperm get made. And this is for any species that has male and female genders. Is that the female gender is purely based on those big food-containing cells. But when you make them, you don't want your food split 4 ways. You want your food consolidated in one cell. And so egg cell production produces one big food-containing cell and then 3 cells that are purely to get rid of the extra chromosomes that are called polar bodies because they originate at the pole of the cell. The division happens right up under the membrane. And a little boop, little cell goes off. Unless you're a dicot plant, nothing happens to that. It's a discard cell. You need to get rid of the extra chromosomes. That's what you do. In dicots, they actually do weird stuff and wind up with a second source of food, which is why the seeds have 2 parts to them. But everything else, it's a discard. In sperm, you got way more of these cells to start with because you got to crank out lots and lots and lots of sperm. Sperm specialize at getting to where the egg cells are. And they have all sorts of ways of doing that. They can swim, they can fly through the air in pollen, they can crawl, roundworms crawl. But the odds are not real good in their favor. You need to crank out and distribute many, many, many of them in order for them to maybe find and get into an egg cell. So, and every time you start one cell dividing, you're going to make 4 functional sperm. So of the many, 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 many more starting cells for males as opposed to females, there also are many, many, many more sperm produced. Because for every starting cell, you get 4 sperm, you only get 1 egg cell. Okay, that is it for this particular chapter.